what we're doing. We're here to start off the project, I suppose. It's going to be running through the semester. And what you're doing, this is your field site for collecting samples. And this is the information that you're going to provide to the City of Sydney Council on biodiversity in this park. So it's around invertebrates. Yesterday, I don't think that was clear to everybody, so people were starting to plan dogs and possums and stuff, and no, we don't have permission. Ethics cuts out a lot of things. Can't do people either. We're not doing plants, so it's invertebrates, okay? So the stuff that you really can't see right now, you're going to do an invertebrate survey of certain parts of the park, not all of them, and Sophie will tell you which bits she's interested in having surveyed and which bits you can't work in for safety issues, or because there's too many members of the public around. You know, we're not going to work in the children's playground, for example. Not a good idea. So, and we'll talk a little bit more about the sampling as we go around. But what you're going to do is, if you haven't already, you're going to be in groups of 4 or 5, possibly 6, but if it's a bigger group, I want more work out of you, so it averages out the same. Well, otherwise it's not really fair. So if you're not in groups, while we're wandering around, find groups, and Matt is a great person for introducing groups to each other. He's really good at this. And then each group is going to come up with an idea, what they want to study. As you listen to Sophie talking about the different areas, the different questions she's interested in, or something else you see, you think about something you want, a question you want to ask about the diversity of the invertebrates. So, you know, why might there be more in one area than another? So Sophie will show you lots of different areas. So you might compare 2 areas. You might compare different sampling methods. If you've looked at the paper, particularly the one I put up from Hitching about rainforests, he did a huge transect up from Queensland up to almost Japan, I think, which is quite ambitious. And he looked at invertebrates in rainforest, which means a huge area of different vegetation. But he was also using different sampling methods to see what you got when you sample differently. So you can have pitfall traps, you can have litter traps, you can have sticky traps, you can have light traps, you can have butterfly nets. There's all sorts of different ways of sampling, and obviously they catch different types of insects, or they catch different areas, things on the ground, things in the air, things, things staying up in the canopy. You won't get them all with one method, probably. So it might be worth comparing a method in a particular area. So have 2 different methods in one area and see what you catch. So there's lots of different ways you can do this, and what we want you to think about today is what you might be interested in. You can always ask us about things that, you know, will they work or not. One of the things is it's a public park, so if you put out expensive equipment or whatever, it may disappear. Cameras tend to disappear really fast. Doesn't matter where you put them, how much you hide them, people find them. So have a think about that. If you're willing to sacrifice some of your samples to the public education or whatever, then that's great. And we can put that into the design maybe. So you will have to think about those things, and within the next week I want you to email to me your ideas on what you want to do, so we can start to sort of negotiate, either ask permission from Sophie whether we can do those things, or is it too big a project, do we have the equipment, how are we going to collect all the data, and is it going to be doable in the time you've got. We'll come and collect the samples, not today, you're not collecting samples today. We'll collect the samples during the Easter break. So if anybody wants to come along and help during that time, it's up to you. It is not compulsory because it's the break, but if anybody's interested, you can come along. And we'll collect all the samples, and they'll be there in the lab for you during the Anzac week when there's no labs going on because of the holiday. So you've got that time free and you can sit in the lab and work on the samples then. So we don't collect them too early and they'll all be dead before you get to see them. So that's the plan. So while you're walking around, get a group together and think about things, write down what you might be interested in doing, and then you can start talking to me on email about what you'll have in place, okay? And then I need an order list. It's like takeaway, you know, you're going to have an order list of what's going to be sampled during the Easter break, and then it'll all be there ready for you after the break. Okay, so that's what we're doing, so you've got to do that while we're walking. So this is Sophie Goldie. Sophie is from the City of Sydney Council, so she's the person who's allowed us to work here. She's the person who's got the questions, that wants some answers from you guys. And Sophie is the urban ecologist. Now the City of Sydney Council is fairly large. I think that the university's in the middle, we're right in the middle of it. So we're part of the area. And Sydney, Sophie is the only ecologist on the council, so there's a huge area to work with and not a lot of help. So this is why people like us are really useful in getting information. So Sophie's going to give you some background about the park and how it works with her job. And then, in amongst all the grass mowing and the planes, we're going to do a quick tour of different areas, depending on the weather, so that you can see the sorts of places you might be. Okay, so Sophie,
yeah. Maybe do you want to come a little bit closer? I just have a really I have a really soft voice, so gather round. Yeah, so it's nice to meet you all. I'm really excited that you're here to start with. This is something that Charlotte and I have been talking about for a while to sort of get students out in the field, in the urban parks, to have a look at what we actually have. For us at the city, we haven't actually done an invertebrate survey in the city of Sydney. We've done sort of little drips and drabs, which has really sparked an interest in understanding what our invertebrates are here in the city of Sydney, because they're such an important, I guess, or they have a really important role in ecosystem health. So this is really exciting that you guys are going to be involved in collecting some of these samples and looking at what we have. Recently we did some research with Dita Hochuli, who's part of the University of Sydney, and he was able to look at a couple of sites in this park and recorded 42 or 24 new species of invertebrates for the Sydney Park. Oh, I can't believe
we should move. Should we move somewhere? Let's move on a bit.
Okay, so this is much better. I don't have to scream at you. So where was I? All right. Great memory. Okay, so yes, so recently in Sydney Park, small research project looking at just some very small areas of unmown grass. We thought we'd heard that might be a good idea to encourage invertebrate species to utilize it as habitat. So we let it grow. Dita looked at it and recorded quite interestingly and relatively high diversity of invertebrate species. We had 4 ant species, 4 butterflies, and I think there were a few other wasps and bees, which was pretty exciting, and we wouldn't have thought that something like that small scale project would actually assist in improving invertebrate fauna species. So that just goes to show for us that even these small scale management ideas or actions, they really do contribute to, I guess, the health of not only the park but also just improves the diversity of species in general. So that's just it. That really excited us about, well, what else is there in this park across different types of sites. And before I go and talk about that more throughout the tour, I wanted to give you a bit of context about this park. Are people familiar about the history of this area? Yeah, I've got a couple, mostly shakes, a little bit of these ones. For those of you that don't know, I'll try and give you a really quick summary of the history of this park. Prior to European settlement, where you were standing at the brick kilns, that would have been a forest of what's called Sydney turpentine ironbark forest, which is now an endangered vegetation community in Sydney. There's not much of it left, and there is obviously none of it left there now. And then down at the southern eastern corner of the park, that was on what's called Botany Sands, and that was made up of, you would have seen swamps and marshes and heathland in that area down that side. So it would have been a really diverse landscape within this area. And importantly, this area, I can't remember the name of the district, but it was known as the kangaroo ground. It was a really important hunting ground for kangaroos for the local Gadigal and Wangal people. And they would have hunted in this area, but also fished and camped in the many rivers and waterways that crisscross this whole area. Down that corner of the park, quite a fair way, there's a channel there called Munai's Channel, which was known, or is now known as Shia's Creek, and that heads out straight into the Botany waterway, so the Cooks River and into Botany Bay. Now that's quite a remarkable history, but then after European settlement in this area, this site was actually turned into extracted for clay for making bricks, basically. So they dug out really big, deep holes, deep pits, and took out a lot of the clay, and then they would have burned them in those kilns to make bricks. So that whole area, most of this park actually was dug out and excavated for a long time, and that started in the late 19th century. I can't remember the exact date, but I can tell you if you're interested later on. So that went on till about the 1970s, so really not that long ago. And also at that time, when it was starting to kind of wind down in terms of extracting a lot of that clay out of the area, in around late 1940s, the brick kilns, not the brick kilns, the deep pits were used as a major municipal waste dumping site, so a tip. So it's gone through extensive changes. It's mainly for household rubbish, but there is a discussion, apparently this is true, that there is an elephant that's been put into these tips somewhere. So it was because of some old circus that used to go around this area. So there's a lot of weird stuff in here. Obviously if you're an archaeologist it would be interesting, but it probably won't get you guys to be digging holes that deep with invertebrates. It would be a bit blah. So that was this place. The tip, I think, finished around late 1970s, 1980, and then it was turned, was it? I can't even remember that either, but it was around the late 20th century, and then a whole lot of soil was basically covering the tip with some building rubble as well as a bit of a capping, and it was then turned into a park. So there's a lot of parks like this across the world actually, where they reclaim land and they build parks on top of old tips. It's not very uncommon. Sounds bizarre when you first think about it, but it's not unusual. So that was around the 1990s, and slowly we've been really improving this park because we know that urban parks are really important for not only our own health and well-being, but also for our urban biodiversity. This is a new project, the wetlands. It used to just be a puddle in some grass. That was all it was. When the park first started, there's been a huge improvement to the system. Basically, just a quick summary, that channel that I was telling you about, that collects all the stormwater from basically the catchment of this area, which is Erskineville, Newtown, St. Peters and so on. There's a lot of water that comes in that channel, and it used to go straight out into Botany Bay. So you can think of all the crap that would go in there and just head out. There was no filtering process. The idea behind this was to capture all that stormwater that went into that channel, send it through this system to get filtered through a number of ways, which I can show you later on. And then a lot of that water is captured and recycled for using in our parks to water our gardens. So we're stopping our reliance on potable water, so the tap water that you and I drink. So rather than using that, we're recycling water. So it's a huge environmental achievement for the council. But also any excess water that has been filtered through this process, also that excess water will go out cleaner into the Botany area. So there's a double positive, a win-win really. So that's really exciting, and I think that what we're seeing and what we hope from this project, that it will improve and increase the habitat for a lot of species as well. We're seeing a lot more smaller birds and generally a lot more bird diversity. So we've got a number of priority species that we're really keen to create habitat for, such as birds, reptiles, frogs. We've got lots of frogs now, especially because of this rain. Microbats too, which we're starting to see more of in this park as well. So it's really exciting, but we don't know much about invertebrates, so that's why it's really exciting to you guys, isn't it? Maybe we'll just start going around and I can show you some of the sites. But did anyone have any questions before we walk away from here? Yep.
I don't have to come up with a question. What kind of questions are you looking at? Sort of, can you give us an example?
Yeah. Oh, yeah. I will do some more.
Oh, okay.
Yeah, I think when you're looking at the site, you can start to sort of ask questions as well. Yeah, but we'll do that throughout the tour. That's easy. Yeah. Sorry.
When you stand for immigration, then you get to quarantine. Big queues. It's much better than it used to be. It used to stand there for hours. But you know, they're searching for everything under the sun. Well, I bring all sorts of stuff in. So I bring seeds and insects. I brought insects in, and I have a permit. And the guy on the desk looks at the insects in the vial, and it looks at the permit and says, "Is this what's on here?" And I go, "Well, I don't know. I can identify them." He can't identify them at 5 in the morning. You know, he's got an extra. So they get taken away to the lab. There's a quarantine lab in Mascot. If you go there, it's stuffed full of everything they've confiscated or they're trying to identify. They identified everything from it. It's fantastic. They're an expert. He was trained by me. The first thing he says, "You taught me in first year." Yeah. And the other thing he says is send more people. We don't have enough people to identify stuff. Everything that comes into the airport, they have to try and identify it. You know, and if it is permissible, then it's allowed in. But otherwise, if you don't identify it, you can't tell. So having experts who can do that, not on the desk in the morning, but in the labs in the background, is a real issue. Finding enough people who have the skills to do that is always really difficult. And it's plants, insects, everything. There's certainly some giant African snails that a little lady, a little old lady, who doesn't speak a word of English, arrived with 2 tied up with strings, still alive, carrying them for dinner that night, and brought them in, and they took them away from her house. I don't know whether she understood the problem because she didn't speak English. But they said, no, they're not coming in. So there are all sorts of interesting jobs in that way. So you could be starting right now. This could be the beginning of your career. Just think about it. You could end up in the airport. They are good jobs. Anyway, let's move on.
Just quickly before we do the risk stuff, if you're involved in the sampling, which I highly encourage, that's something that I would love to be doing rather than being at my desk and managing.
Well, you're invited, so.
Yes, I am invited, so I'll be there. But some of the things that, like you're working in an urban park, and so some of the things that you should be really aware of, firstly and foremost, are the park users. They're really interested and they have a lot of ownership over this park. They're really attached to how it's been improving, and you'll probably, even yesterday when we were out here, somebody told us to keep the gate closed, and so they will be vocal about anything that they see that's not kosher. So basically, if people do come and inquire about what you're doing, or if you're around here and you may look suspicious, you're obviously with notebooks and looking at things on the ground or in the water, who knows, then just, if they come up and ask you what you're doing, just let them know. Tell them that the city knows about what you're doing, the City of Sydney Council, and they'll probably more than likely be really interested in what you're finding. They really are, curious, but as I said, they really do feel attached to this park. The other thing too is that often you see, and you probably have seen today, is that there are a lot of dogs in this park. They're not a risk per se, but just, it's always good to be reminded that, there may be 1 or 2 that aren't that friendly. So I'm a dog lover, and I have been guilty of going up to every dog I see to pet it. But I have had a few bites in my time as well. So that's now a pastime for me, and it just really encourage you also, if there are any dog lovers, just be wary that there are dogs that aren't so great. That's just something I do have to reiterate. The other thing is, if you're working in some of the sites that I'll show you this morning, again, it's an urban park. You get a lot of diverse people using it during the day and night. And so you might find a lot of sharps, broken glass, other things that can really pose a risk to you if It's, remember, if you are, there is a possibility to use, to sample water at a couple of points in the park, not throughout the whole wetlands, but reminding you all that the water quality isn't that great, so again, gloves will be needed too. So it's just, and also gumboots, but I probably won't be advising you to wade through it from the water. No wading. Yeah, so forget about that. Let's start walking.
No swimming.
Yeah, no swimming. No sailing. Okay. So this is one of the sites that I was talking to Charlotte about. This cascade area adjacent to our land site. This site, as I was talking about the wetland project and the new kind of stormwater harvesting, a lot of plants were garden beds were part of that project. So there's thousands of meters squared of additional garden beds that were probably planted maybe a year and a half ago, approximately. So quite new. So for that reason alone, that's quite interesting for us. Like, what is the diversity of species now in this site? Is there a lot? Is there a little? Are there a dominant species of one or not? What's on the ground in the mulch? Is that different to areas that don't have mulch, which is adjacent? This doesn't have a hugely, well, it's not covered by a lot of these canopy trees up here, the acacias and gums. So the focus really was on lifting that mid-story layer of vegetation. It's in the growth period now and it will grow, but at the moment it's not as high as this site over here. And this site doesn't have as much mid-story or ground cover plant species. So are they different? What's better? What's worse? There's a whole lot of questions that are in our head about this. I was saying yesterday too that a lady who studies native bees and solitary bees in particular, she was telling us that putting mulch down on garden beds is something, it's a practice that may be not so great for those solitary bees that are actually looking for places to nest in the ground because they can't go through some of that dense mulch. But it's a standard practice for council to do in a lot of parks, but we don't know whether or not that's actually, we don't have really hard evidence to show that we do have native solitary bees in these areas.
Where do you get your mulch from then? Is it made on site and used here, or would you bring it from somewhere else?
It's mostly from trees that we've had to remove within the City of Sydney council area. So we mulch them, yeah, here.
So you wouldn't be bringing stuff in from outside which might bring other insects or inverts in?
They not, well, yeah, they could, but they're outside of this park, yep, but yeah.
One of the things that strikes me, this is quite a good place to talk about the different sampling methods, because you can look in terms of the mulch, you know, you could collect mulch from different areas and look and see what's in it. So that's like leaf litter. There's not a lot of leaf litter around there, but there's probably enough. It's fairly sparse. So you can do leaf litter sampling fairly easily, and you could do quite a few of them. You want to think about replicating what you're doing, you don't just take one. But obviously not everything is living down on the ground. We can do soil sampling as well, which is different to leaf litter. We've got various ways you can sample inverts in the soil. You can use Colburn funnels, which push everything down into sorting trays. But a lot of the things may be living in the vegetation. So, as Sophie said, the vegetation here is mainly undergrowth. So you can, how you get what's living in there, maybe butterfly nets. I'm never quite sure what butterfly nets are doing, but you could try it. What you could do is compare two different sampling methods within this area. So try nets and maybe sticky traps. Now we had a big debate about sticky traps yesterday. They don't do well in the rain, obviously. Nothing does. And inverts are not going to be around so much in the rain. But and they do get very sticky, so it's rather a messy way to work, but You could catch different things with nets and with sticky traps. Obviously, if you leave any sort of equipment out here for a period of time, the chances are that visiting things may come and visit and take them, or I don't quite know what dogs do with sticky traps. Could be interesting. So you get bits of little nose impressions on your traps. Could be fun. The other thing is you could hang sticky traps in the trees. Hopefully people won't come and collect them. You'll have to think about how long you would leave them. The other thing you can do, light traps, I don't know that we're going to be doing a lot overnight, so that may not be one way of doing it. But you could compare areas, you could compare same sampling in there and in here with the differences in the understory and the lack of canopy. Or you could look at two different methods within the same area. And you can do this for all the different areas around the park. Don't make things too big. But something that's a comparison will be quite interesting to look at, because then you can start to think about why it might be happening, which is the sort of questions that Sophie's asking. Is what we're doing here working? In which case maybe we'll do more of it. If it doesn't work, maybe we'll try something different. So if we can all be sampling different types of areas with different types of sampling methods, we can cover a lot in a fairly short time. So have a think about that as well. So this is two really interesting areas with different sort of histories, and obviously you would think different inhabitants, but maybe not. It may be that some particular groups of inverts don't care and they equally inhabit this and that quite happily. So it could be you target certain groups, but again it's hard to know which ones. If you read about something that is, well, threatened species are difficult because usually there's not many of them, that's why they're threatened, so finding them is difficult. But something that's a key species in terms of environmental health is often something you can go searching for. But you may need a lot more samples then to make sure you're getting good information. So again, have a think about that, and we'll move on to another one.
Of this hill, you can see that we've done some recent plantings, and we've done a reconstruction of eastern suburbs Banksia scrub, another endangered ecological community. We'll probably ask Matt actually how the percentage that's left. Matt, do you remember recall the percentage of eastern suburbs Banksia scrub in Sydney?
What's left?
Yeah.
Nothing.
Yeah.
1 to 2% or something. We used to cover that whole area from North Head, like South Head and all over, and now it's about 1%.
Yeah.
Mainly on golf courses.
Yeah. So there's only a really small amount of original vegetation left. This is not original vegetation. Like I said, this was a brick kind of excavation, creation, manufacturing site that had the deep pits, and then we had the tip on top of that, and then it's a park. So the idea behind reconstructing it it had two reasons: to create awareness about the original landscape of the area, but also to create habitat for some of our priority species like the reptiles, the blue tongues, this is really one of the main areas that they're left within the City of Sydney local government area, but also things like the small birds, our fairy wrens, our blue fairy wrens that you might know. There's not many of them left within the City of Sydney local government area, and other small birds and so on. So the idea is to create a lot more of these sites that have these dense mid-story areas. This was also a site that Dita from University of Sydney had a look at and found some interesting things compared to the unknown lawn that he also had a look at. Interestingly, in this site there was only one ant species, and that was an invasive species, the Argentine ant. Why that is, we have no idea. Is there something that we're doing wrong in this area? I don't know if you can say it's wrong, but what is it about that site that's different to the site over there? So if you do compare sites, these are some of the questions that you could think about. It might be something to do with the substrate that we use. We've used crushed sandstone to plant the native plants into. That's a really good medium, because the native plants If you know, they don't like a lot of nutrients, and a lot of these plants are on coastal areas that that do grow in sandstone-rich areas. So now that you can see, there's quite an edge to the area that we've planted. One of our goals is to lift that mid-story under these canopy trees. So we're slowly coming through. This area was planted about 2 years ago. 2.5 years ago. I have to look at, it was National Tree Day in July 2015. Yes. So there's been a lot of growth there, but you can see that the plants that were planted under the trees have sort of low growth compared to up on the hill. But you can also see that it has a lot more leaf litter. You'll notice that compared to the eastern suburbs Banksia scrub site. And within the Banksia scrub site, we've actually put a lot of these logs in here that some of these are trees that we've had to remove from the council area. And rather than turn them into mulch, we're trying to replicate some things that you would see in nature. So we'd leave a lot of fallen trees. We'd put these logs in to act as habitat for our blue-tongued lizards, for instance. And what's curious to me is the idea also behind it is, will they improve the soil health as well after it breaks down? So is there a lot more invertebrates around these logs? Do they spread out a certain distance around those logs? I don't know. I'm hoping you guys will find out. If you're interested in looking at that. Also, this site is interesting because we have a creek that runs down. It's generally wet most of the time, but if we do get a dry spell, it will dry up. But who knows? It doesn't look like it's going to let off this rain for a while. So that's also a really interesting site because we are seeing frogs up there. We know we get frogs down there. What are they eating? Is there enough foraging resources for some of these animals? And does it really differ a lot between the edge of the creek and say a site around here? So there's some of the things, that just come to mind about if you really are looking at the diversity of invertebrates, particularly at this site. So it's really interesting. I think it's interesting.
Can I ask, how old are these trees?
Good question. So they're not that old. When the park was first created, the idea for planting out areas was just to plant trees. So this is an old style in a way. We're trying to do it differently by planting that mid-story out as well. But the park was created early 1990s. The trees here don't get that big because it's a really different site, obviously. If you're thinking about the fact that it is on the landfill site, there is a relatively small capping of soil that's on top of that. So it would be stunted with growth, but I would say anywhere between 10 to 20 years. Yeah. Obviously the casuarinas, they're small because they just grow from suckers. Yeah, and they're quite young. But yeah, we do have some large ones. This site is a National Tree Day site as well. It was planted 2013 and 2014. So it's a little bit older than that site up there. You can tell just by the size of some of these trees that they're also a little bit older. It's a little bit more diverse with that with the canopy and the understory plantings compared to the eastern suburb sites and just the pure kind of stands of canopy trees. So there's some differences in that to consider. This site hasn't been surveyed by DETA that I was telling you about, so we don't know much about this site. We don't know how many ant species are here. We don't know really much about anything for here. But also interestingly, we have another creek down this side, and it's got a lot of weeds. So thinking about what's in there, what invertebrates like in the weed section, are they different to a re-veg section? point we do have them all fenced, well, loosely fenced. That's to stop a lot of people trampling through. The eastern suburbs, Banksy Scrub, is we're quite protective of that site in a way from people going through. So if you are working in that site, that's just something to consider, being really sensitive about some of those plants, because some of them are actually really hard for us to get. It took about a year to order quite a few of those species up there. This site, I still care about it, but it's that sounds terrible. But we just we don't have as many sensitive plants here as well. But we have fenced it off to stop people just trampling all the way through. But you can enter there if you want to do some surveys there, and also up there. That's just something to be mindful about as well. Otherwise, I would never invite you again. No.
It's a really good leaf litter.
Yeah, absolutely.
And the weeds are wonderful. So it would be good this one.
But
it's a much more dense canopy all the way up. There's the layers are much more obvious here rather than gaps between them, which is interesting. There's a bit of a monoculture going on over here with the casuarina, which is nice. Because you notice where the casuarinas are, all of this lot. You don't get anything growing underneath them. It's such a thick layer of needles or whatever there. So whether that affects the invertebrates there would be interesting, because here you've got more diversity of types of leaves. The structure of the leaf litter is different there to here. You know, you think leaf litter is leaf litter, but in fact that's very different. It may retain more water. I don't know if they need that. It doesn't dry out. Temperatures, etc., could be different. So that's actually quite interesting, that bit. I hadn't thought about it.
Yeah, that's true.
So
I think, yeah, so that's what
I'm not interested.
Okay. Of one species. And this is lady grass is quite a good grass because it's quite resilient, it's quite hardy, it spreads through rhizomes, so it can really dense up a site. But it's interesting because it is so different to some of our re-veg sites. It's different to our woodland sites, but still, you saw there was like so that's interesting for us. Like we do these mass plantings, but is that something that it might not be an objective to increase invertebrate species in there, but by default, what is the invertebrate diversity like in some of these? You may be a bit surprised by finding a lot of different species of ants or insect pollinators. We don't know. It's just we have this dearth of information about some of these sites. So that's a pretty basic site, and you can see it's right up against an exotic grass lawn that's typical of a lot of these urban parks. So are those two sites different, or are they the same? That's pretty much all I had to say about that site.
That's a good question.
Sites that I've shown you, we do occasionally have volunteers working, doing some bush regeneration volunteering, which you can also get involved in. I can send Charlotte the link. But also we'll have some staff that will be doing a lot of weeding across these sites. So it may not impact some of the work that you guys are doing, but just to know that when you do go into those sites, you will also come across these people that they probably want to know what you're doing as well. All right. So this is the last site. We call this one of our bioretention swales. Does anybody know much about those? Have they heard of that phrase before? No? So when I was telling you about the stormwater that was being collected from that channel just up in that corner, one of the processes of making it cleaner, of filtering it, is that that stormwater will run through these systems. The way they work is that these certain plant species that have been planted in here have been researched quite extensively in their role at accumulating some of the rubbish in the water, some of other toxins, and they're really effective at doing that. We have to replace them every 20 years or so. But this planting has been, it's a year old, year and a half old, and they love these sort of situations because the stormwater, if you think about all that stuff that comes off the area that we utilise, it's full of nutrients such as phosphates and nitrates, and these plants, they love it. And that's also why you see quite a lot of weeds in here as well. We're just getting an understanding of the maintenance requirements for these sites, but I won't go into that today. But you'll see there is a lot of thick growth, and you'll see as Charlotte pointed out, there's of plantings of certain species. That's just the way they've been designed. And this site is obviously quite different to some of these sites. They'll be a little bit wetter. We're not really, we don't want you guys to get too close to the water's edge, so it'll just be up the top here. There's a pathway down the bottom and then some plantings. We won't want you guys to go in that section. But the cascades that you saw that were further up, we walked past them with the stepping stones through there. You can do some water samples up there if, say, you wanted to compare that site to some of these creek areas, or if you wanted to look at, say, the muds of that cascade area, the soils, I should say, compared to this site. It's one of the questions that I'm curious about. One of our priority species that I was telling you about, birds, small birds, but also wetland birds and waders, things like royal spoonbills, cormorants, what else do we have? Like black-winged stilts, I'm not sure if you know those guys, they're beautiful birds, small dottles. They like to forage in sort of the shallows of wetlands, and what I'm interested in is for some of those birds, not all of them, do they have enough foraging resources in the wetter soils that they can utilise? We don't know what's in there. At the moment we don't see many of those birds here since we've done these plantings, and that's for other reasons. But we're just curious about what is in that soil, if there's much at all. Is there one species or is there a diversity? Looking for a diversity, which is great. There's quite a lot of resources out there to increase your understanding about water invertebrates, but also I don't know how much resources are out there around the soil invertebrates. That's like Charlotte said. Yeah. So this is a really interesting site for us. We then also have some logs and rock piles over there as well, and frogs definitely utilise this site too. If you come at night time.
Yeah. You could sample around the logs, near the logs and away from the logs.
Yeah. And the other thing that we do in this park, say monthly, bi-monthly, is do some microbat detection. So we walk around with these little microbat detectors, and we try and, we can't see them, but we can hear them when we use these detectors. This site and the site just above the eastern suburbs Banksy Scrub, that's where we seem to get more activity of microbats foraging. So that tells us that there is quite a lot of invertebrates flying around for them to be able to utilise. And seasonally it does change. We've probably been monitoring it for a few years now, so we have kind of varying sort of insights into when certain species are around and when they're not. But I'm really curious about, but as Charlotte said, I'm not sure about whether or not you can do anything at night, but there may be something that you could leave overnight that's inconspicuous, and then you could come and possibly collect in the morning to see what is around in the evening. Possibly. So that's really interesting. You'd have to negotiate that. And sort of comparing those sites to, it might be possible to look at sites that have more light pollution compared to sites that are a little bit more darker. But again, have to negotiate that depending on how you would sample and considering that's nighttime. Night sampling. Yeah.
So you're getting the idea now of what sort of things you could do. So I think a lot of it doesn't have to be a comparison, but I think a comparison would be good, just so that it gets you to think a bit more about what's happening in different sites or with different sampling. So leaving things overnight, we'd probably need a bit more help with that, but we could work on that. That's why what I need from you guys within the next week is, an email which gives the people who are in the group, so I know everybody's in a group and doing something, and then an idea of what you might want to do, so the site or the sampling or whatever, just so that we can get them all together and see the enormity of what we've docked ourselves in for during the break. And then how much help we might need, because I can get other people to come and help sample. But if you want to come along, it would be really great. So as soon as we see that, then we can negotiate whether we can do things. We may have to come and ask Sophie, can we go to certain areas, or what can we sample, what can't we? We don't all want to be doing the same thing because we'll just wreck the whole place. So we do want to spread this problem around the park, but also in the different habitats. Just so we sample more, but also we don't take away all the invertebrates and destroy any diversity that they have. So we don't want to have a lowering of diversity just because we've done a survey, which would be a bit sad. So, is anything else you want to say? I think that's everything. Has anybody got any questions? Not right now? Yeah.
We give you like a sample area to look at. Will you help us decide on like a sample method? Or do you
you have a think about it, put an idea to me, and then I can help you to work it through. Yeah, oh definitely, that's the whole point. So if you send me something, if I don't have anything, I can't work on it. Once you send me something, then we'll set up, I think Matt's going to do it, we'll set up a piazza site just for the project, so you can discuss amongst yourselves. But we can also, might be able to set up groups in Blackboard, or just email me, and we'll discuss with each group or with the whole team, so we're not all doing exactly the same thing. And some things we may not have the equipment, or some things, if you set things up, if it's bad weather you won't get anything, or things will disappear. You put anything up, people seem to find them, or people's dogs find them. They're like magnets. It's just unbelievable what people can spot. They walk through and they see absolutely nothing, but they will see your equipment or your tiny little sample, and they'll zoom in on it. So we take that into account. Working in urban areas, you have to accept that. So we'll plan that, but you need to be thinking about the numbers of replicates. How many samples you're going to take. That may depend on how many people in your group. If there's more people, you can clearly do more work. What you'll do is we'll collect them, and then you'll have the lab class, the ANZAC week, you'll have that morning, in your case, to do the sorting and to come up with your data set. And then we can sit and work with you on that as well. And then you can start to think about how you might present it. And we can talk then about videos. So if you don't want to do videos, you want to do something else. If you want to do a dance, you can do a dance. I don't really mind, as long as the videos anyway. So, but and then we'll work on that along to the end of the semester. And then you're going to hand it. I can't remember when you're handing it in. Is it week 11 or something? Week 12, yeah. And then we'll have to have a get together. Probably, evening or something, or late afternoon, when we get everybody together and we can see what we've all got. And we have to mark them all as well. But we haven't got that far yet. Let's just work